The smell of ripe fruit could halt the growth of cancer cells, scientists say. Ever since the COVID-19 pandemic kicked off, we've been far more aware of our sense of smell. Now, new research shows that odors like those emanating from ripening fruits or fermented foods could lead to changes in how genes are expressed inside cells far beyond the nose. The findings have scientists wondering if, with much more research, sniffing volatile airborne compounds could be a way to treat cancer or slow neurodegenerative diseases. While the idea of delivering medicine via the nose is not some new idea, that's of course a huge leap to make. From experiments in cells, flies, and mice, there may also be unforeseen health risks associated with the compounds tested, so more studies are needed to better understand the downstream consequences of this intriguing discovery. That exposure to an odorant can directly alter the expression of genes, even in tissues that have no odorant receptors, came as a complete surprise, said Ananda Sankar-Ray, a cell and molecular biologist at the University of California, UC Riverside, and senior author of the study. The team exposed fruit flies, Drosophila melanogaster, and mice to different doses of diacetyl vapors for 5 days. Diacetyl is a volatile compound released by yeast in fermenting fruit. It was historically used to impart a buttery-like aroma in foods like popcorn and is sometimes present in e-cigarettes. It's also a byproduct of brewing. In lab-grown human cells, the team found diacetyl can act as a histone diacetylase, HDAC inhibitor. It triggered wide changes in gene expression in flies and mice, including in the cells of the animal's brains, the mice's lungs, and the fly's antenna. HDACs are enzymes that help wrap DNA more tightly around histones, so if they are inhibited, genes can be expressed more readily. HDAC inhibitors are already being used as treatments for blood cancer. In subsequent experiments, the researchers found that diacetyl vapors halted the growth of human neuroblastoma cells grown in a dish. Exposure also slowed the progression of neurodegeneration in fly model of Huntington's disease. Our important finding is that some volatile components emitted from microbes in food can alter epigenetic states in neurons and other eukaryotic cells, Ray says. Ours is the first report of common volatiles behaving in this way. The team studied the effects of diacetyl as a proof of concept, but given other research shows that inhaling diacetyl causes changes in airway cells and even a lung disease called obliterative bronchiolitis, a popcorn lung. This compound may be not be the effect of the perfect candidate for therapy, Ray admits. We're already working on identifying other volatiles that lead to changes in gene expression. Ray, who has found 2 startup companies and filed several patents based on his team's work, there are lots of other limitations to the work too. As one peer reviewer of the study points out, the study falls short in providing a thorough analysis of the underlying mechanisms that could explain how odors induce epigenetic changes in cells a long way from the nose. Whether the prolonged or repeated exposure to commonly encountered odorants may have longer-term consequences was also beyond the scope of this lab-based preclinical study. Remember, as the saying goes, it's the dose that makes the poison. Given our repeated exposure to particular flavors and fragrances, the findings outlined here highlight a new consideration for evaluating the safety of certain volatile chemicals that can cross the cell membrane. Perhaps a more practical application of this work is in agriculture, as plants also contain HDAC enzymes, and other research has shown they exhibit a strong and sudden response to volatile chemicals in the air. The study has been published in eLife, and this is on Science Alert by Claire Watson. Please leave your comments, and thank you for your support. Kindly support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.